Hello, ladies and gentlemen. Our project is development of a model for predicting base sequence mutation rate in the COVID-19 virus using the neural language processing technique. Mutations in COVID-19 are caused by abnormal division of the base sequence in the DNA. The host environment can trigger these events. There are three main methods of mutation. 1. Change the original sequence to a new one. 2. Create a new base sequence. 3. Delete the base sequence. We will apply long short-term memory as one of the AI methods. It's by usually used to predict the next word, but our project uses it with DNA to analyze upcoming mutations. Our task is to develop a model that predicts mutation from the long short-term memory method. Objective 1. To experiment to develop a model for predicting COVID mutation. 2. To study the efficiencies of the model from using the batch size. 3. To study the results of a comparison of the learning algorithm model. 4. To study the efficiency of using convolutional neural network with long short-term memory in different layer positions. And 5. To classify the severity of the virus using the classification model.
Next is the preparation of the datasets. Initially, we will collect the sequence dataset for all three strains of COVID-19, Alpha, Beta, and Delta of the Philippines from the KISS and next-gen site. Next, the dataset is aligned so that all bases are the same site, and when the alignment is complete, a total of the 300 sequence of data will obtain, divided into the Alpha, Beta, and Delta 100 sequence each, with a size of 29,992 sequence. Next, we will analyze all these 300 sequences to determine the point of mutation. Once we find the mutation point, we will take this data to the data model first, and then we will divide it into the training set and the test set in 80-20 ratio. Next, we will change the base order in which the mutation occurs as the number 0 and is stored in the data classification dataset, then use the data model for processing. So we will adjust the letter A, T, C, G to uppercase and adjust R, K, M, N, Y, S, W, D, B dash to 0. Then we will convert the dataset from the previous step to KEMA and tokenization to adjust the format information to be ready continue training the model.
The principle of the model is taking a sequence of 9 bits, store the data encoding, and then having the model compute reverse to get the next DNA. Next, we take one sequence from the data classification as the starting data for the model to predict. It finds the point 0 and use the 9 predicting that DNA to make the model predict the next base and replace it with 0. So we get a new sequence, so make the sequence more accurate. We need to adjust the model to have a low loss, which is in step 3. The lab tests are all implemented. Here's the loss graph of our models. It can be seen that the model is well learned and the model can learn much more. Result architecture conveniently worked by our team is the best model.
Next, we will create a classification model to classify the severity of COVID-19. By classification, it is divided into three classes, namely alpha, beta, and delta. We have studied the further that the virus then affect the severity of the virus as well, with beta being the least severity, followed by alpha and delta most severity. Here is an example of the base sequence obtained by applying the sequence generation model to predict the location of the mutation in the base circle as shown in the figures. When the classification model takes the set sequence after predicting it turns out that the base sequence is 98% close to the delta genetic. In the next step, it is simulate protein structures comparing to the base sequence that we have generated with the Wuhan character as shown in the pictures. And here is the comparative pictures between normal viral and the viral sequencing generated by the model. From this result, we can use the data to analyze in many ways, such as refer to the structure of the virus or look at the trend of the virus mutation behavior.